White blood cells and the immune system. So, what are white blood cells? White blood cells, also known as leukocytes, are the only formed elements in blood that are complete cells with nuclei and organelles. They play a crucial role in defense against disease and function as a part of the immune system. What is the immune system? The immune system is a defense system that protects the body against foreign invaders such as bacteria, viruses, toxins, and cancer cells. It is composed of immune cells, lymphoid organs, and tissues that work together to recognize and eliminate threats. Let's compare humoral and cellular immunity. Humoral immunity is controlled by B cells, which produce antibodies. When B cells detect an antigen, they become plasma cells that release antibodies. These antibodies circle in the blood and lymph, bonding to bacteria, viruses, and toxins. This marks the pathogens for destruction by immune cells. Humoral immunity works best against threats found in body fluids. Cellular immunity is driven by T cells, which target infected or abnormal cells. Instead of making antibodies, T cells either kill infected cells directly or help coordinate their immune response. Cytotoxic T cells destroy infected or cancerous cells, while helper T cells activate other immune cells. This type of immunity is essential for fighting infections inside cells, like viruses and some bacteria. Let's compare and contrast the origin, maturation site, and general functions of white blood cells involved in the adaptive immune response. B and T lymphocytes are key white blood cells in adaptive immunity. Both originate from bone marrow stem cells, but B cells mature in the bone marrow while T cells mature in the thymus. B cells control humoral immunity, producing antibodies that mark pathogens in body fluids for destruction. T cells drive cellular immunity, with the cytotoxic T cells killing infected or cancerous cells, and helper T cells activate other immune cells. Now let's define and describe immunogenicity and reactivity with respect to the adaptive immune system. Immunogenicity is an antigen's ability to trigger an immune response, activating B and T cells to produce antibodies or immune reactions. Reactivity is an antigen's ability to bind to immune components like antibodies or receptors, even if it doesn't fully activate an immune response. Key differences are immunogenicity activates the immune system, reactivity binds to immune molecules but may not trigger a full response. Let's describe and define the process of becoming an antigen-presenting cell. An antigen-presenting cell, or APC, is an immune cell that shows pieces of a pathogen to T cells, helping to start an immune response. First, cells like macrophages and dendritic cells engulf foreign substances by surrounding them. Inside the cell, the pathogen is broken down into small pieces. These pieces then attach to major histocompatibility complex, or MCH molecules. Next, the antigen MHC complex moves to the cell surface where it is displayed. T cells, especially T helper cells, recognize these complexes and become activated. This activation leads to a broader immune response, including the activation of other cells such as B cells and cytotoxic T cells. Lastly, let's describe active and passive humoral immunity. Active humoral immunity occurs when the body makes its own antibodies in response to an antigen, providing long-term protection due to memory formation. Naturally, happens after infection. Artificially, occurs through vaccines that stimulate antibody production. Passive humoral immunity happens when antibodies are received from another source, offering short-term protection without memory cell formation. Naturally, antibodies pass from mother to baby through the placenta or breast milk. Artificially, antibody injections. Resources for this video are Merib E. and Hohen K. 2019 Human Anatomy and Physiology 11th edition.